Welcome everyone. Thank you very much for joining today for our webinar. We really appreciate that you are taking the time to listen to us and to see what updates we have in the oncology genetic testing. As you know, the title of the session is NGS in Oncology, Bridging Molecular Biology and Clinical Practice, which is quite self-explanatory. As most of you in the audience know, our oncology division specializes in tests for somatic mutations with reports which intend to be as useful as possible for the precision treatment for clinicians. And today we will present the newest tests we developed with the multiple biomarkers we cover in the different tests. And we will have a special focus today on the HRD analysis due to its importance in ovarian cancer. As you know, we have a proven correlation with the myriads my choice, which is great. And now, in interest of time, let me just introduce our speaker today, who is Vanessa Rivero. She's a biologist with a master's degree in clinical cancer biology. She's currently the head of our oncology division at Bioarray, where she applies her expertise in molecular biology, next-generation sequencing, and data analysis. Throughout her time in multiple laboratories, she has gained experience in cell culture, DNA, and RNA extraction, as well as methodologies applied to molecular genetics, always focusing on the field of oncology. So without further ado, Vanessa, please, you can proceed. The floor is yours.
Thank you, Luis, for the kind introduction, and thank you to everyone who is now connected to hear what we have to say in Bioarray. And I, first of all, would like to show you the different parts that the seminar we are here today to hear is going to have. First of all, I would like to give you a small introduction in which I will tell you why it's so important what we do. Afterwards, I will go through the solid tumor testing and making a focus on the HRD testing, as Luis just mentioned. We will also talk about liquid biopsies, and I will take a couple of minutes of your time to tell you some things about hereditary cancer syndromes that we do not perform in my department, but we do in Bioarray, and it might be of your interest. So, first of all, we have the little introduction that I want to give you. I'm going to tell you something you already know, I'm sure of it. Cancer is, after all, a disease of the genes, where genetic alterations on genes that control how cells multiply and divide are affected. And we do have this disease that actually affects 1 in 5 people in their lifetime. I mean, 1 in 5 people that you know will develop probably cancer among all the years that they live. So it's quite interesting to know more about this disease. 1 in 9 men will die from it, and 1 in 12 women will die from it, which is a higher rate than other diseases, for example. All this data is extracted from the World Health Organization, and it makes sense that we have a global effort going on to try to develop more accurate treatments and drugs that help all these patients that suffer from cancer, and to give them to the correct patients. And that's when Bioarray comes in. From only 2 blood samples or FPPE tissue, we are able to tell you which are the treatments, drugs, or in case that there are no treatments available, we tell you which clinical trials your patients might benefit from. We process the samples, we extract DNA, RNA, maybe both, depending on the test that I will later get on more explanation about it. We sequence it, and we give you the information of the different mutations that your patient has. With that information, we also provide some information about which is the best treatment option according to different globally accepted companies, associations, etc. And also, as I just mentioned, different information about clinical trials that your patient might fit into it. I do think that the best way for me to explain what we do is definitely go into all different tests. For that, I'm going to start with solid tumor testing on FFPE samples. and with the main part of the presentation today, which is HRD. As mentioned, is a quite relevant testing for the nowadays treatments available. So when you have a patient that don't really know what to give them and they have ovarian cancer or something similar, if they have the HR pathway affected, they might benefit from PARP inhibitors. What we do here is we evaluate 46 different genes in this homologous recombination repair pathway in solid tumors, of course, to detect if they do have this deficiency. I mean, the result that we give you is that if they are HRD deficiency positive or negative. We also include some other information such as genomic instability metrics, and all this information we obtained for the sequencing of these genes that I told you, and from only 20 nanograms of input of DNA extracted from FFP tissue per reaction. We only need that small amount of DNA to be able to help you with the diagnosis and treatment of your patient. If this is not enough for you and you want something else, we do have this huge panel that is one of our biggest ones, the biggest one, in fact, in which we include over 500 genes. Both DNA and RNA testing are applied in this case. We also need only 20 nanograms of DNA and RNA extracted from solid tumors. And what we will see is different single gene biomarkers and multiple gene biomarkers. We are going to see mutations, both in single nucleotide variants and indels. We have novel and null fusions and a long etc. that you can see in the screen right now. What you will get from this is both all the information that I just told you and also the information about the HRD testing. You will have the value of positivity or negativity of the HRD deficiency. So if you really know that you don't want to test the HRD pathway, we have also the Pan-Cancer Comprehensive Assay Plus, in which we do include all the genes that I just told you, all the single gene biomarkers, the multiple gene biomarkers, and both from DNA and RNA. In this case, we do not include the information about HRD, but we do have a lot of relevant information and we will help you treat your patient. After all, that's the objective of us all. The thing about this is that I think the best way for you to see what information you will obtain in testing your patients with us is to give you a sample of a report that we make. So in this case, we have a sample of FFP tissue that we tested with the panel that the assay that I just presented. And this is a breast cancer patient, and we find one relevant mutation that you can see in this table. You have the relevant breast cancer findings according to the test that we are performing. We, of course, give you more information about the biomarkers that we just found. We tell you the genomic alteration, the relevant therapies in these cancer types, or including other cancer types too. And we give you the information about the clinical trials available today. Of course, we will give you all the information that we have about the DNA sequence variants, including the amino acid change, its coding, etc., etc. We have why is gene important in the case that I'm trying to treat the patient with, and you will have potential relevance, the alterations that are frequently found, and also the background. You will see how there are also some relevant therapy details in which you have FDA information, NCCN information, EMA information, and ESMO information. All this information will help you with the treatment of your patients. These are all institutions that we all know and that we trust for patient treatment. As I mentioned, in this case, it might not be as relevant because we do have some option treatments that are relevant for this patient. But in the case that there are none, or even if there are cases in which it is relevant, we give you the information of the current clinical trials in which this patient might benefit from. Now, we are going to start to go down in the number of genes that we are testing, but we are always going to try to have all this information that I just told you with an accurate diagnosis, selection of best fitting treatment, patient selection for clinical trials, and increase on survival rate. In this case, we're talking about the pan-cancer comprehensive assay, in which we have less genes than in the other cases that I just presented to you, but we do, even though, cover 161 genes. These genes are all relevant in cancer in different solid tumors, and that's why we have included them in this test. We are also going to test both DNA and RNA from FFPE samples. We only need 10 nanograms of DNA and RNA, which is a really low quantity if you think about it. And we include a lot of information, including gene fusions, copy number variation, etc. This is a bigger panel comparing to the next one that we are going to talk about. That's the target assay. In the case of the target assay, we are going to try and focus on 52 different genes. We are going to be testing for hotspots, copy number genes, and fusions. So we are going to need DNA and RNA again from FFPE. This might be of special relevance if you have a suspected gene you want to test and you want to have other options in case the gene that you thought firstly would be the one with the treatment, you don't have an option to treat your patient with it. So you have a wide variety of genes that are tested. Of course, always we will give you the information about the different treatments, the different possible clinical trials, etc. And now we are going to focus on the smaller assays that we offer for this kind of sample. And we are going to talk about the colorectal cancer test. In this case, we are testing for 19 different genes. Some of them are the genes of the mismatch repair pathway that we all know are of high relevance in this kind of cancer. But we do not stop there. We have other relevant genes that you will find really interesting when testing for this kind of cancer. And in this case, we have two different samples possible. We will need an FFPE sample, as I have mentioned in all the assays that I just talked to you about, but we can use a blood sample in the case that you want to test for a germline mutation. Once again, we are going to include 19 genes that are of great relevance in this kind of cancer and as single nucleotide variants, indels, and copy number variations. It is true that we do not include RNA in this test, but we do include DNA and a lot of relevant genes for your patient's treatment. If now we talk about lung cancer, one of the biggest threats when it comes to cancer, we have this panel of 12 different genes in which we can see again genes that we all have come to know as really relevant in this type of cancer, such as ALK, BRAF, EGFR, etc. But in this case, we are going to see fusion genes, which do have targeted treatments for this kind of cancer, as you all know. And we will see how with 20 nanograms of DNA and RNA extracted from FFPE tissue, we are going to obtain all the relevant information that I just mentioned. In the case of prostate cancer, breast cancer, etc., we do have a test that's called the BRCA test, the BRCA extended test in this case. It does include 21 genes that provide information and pathogenic variants in these genes, along with genes recommended by the Spanish Society of Medical Oncology. These genes are also related to the homologous recombination pathway that we did mention before, and we all know the high impact that they have in patient treatment. So it would be a great option for your patients too. In the case of our smallest test when it comes to FFPE testing, we have the BRCA test, in which you can see that we do include two different genes, the BRCA1 and BRCA2, and we do include different information extracted from DNA. Now that we have covered all FFPE information, I would like to go into the different liquid biopsy tests that we have available. And as you know, liquid biopsy is becoming every day, bigger and more important test when it comes to oncological patients, as we have seen how these tests are quite non-invasive. You have a lot of information, and we have a great resolution and limits of detection that might help treat your patients and be aware of the possibility of a new oncological process to be going on. So I would like to start with a bigger panel, which is the pan-cancer assay. We do include 52 genes, in which we only need 2 blood samples from which we will extract ctDNA and RNA. We test both DNA and RNA from these samples. And once again, everything we have is for actionable variants. We are not testing just because of the pleasure of testing. We want to help your patients. So we do have really relevant genes tested here that have approved labels, are included in guidelines, and do have relevance in clinical trials. As I mentioned before, the detection limit of this kind of test is quite low, as we have a 0.1% of detection in relevant genes in solid tumors. Once again, we do also have different options for those panels that are more focused on different kinds of cancer, and that's on the colorectal cancer, for example. We have 14 different genes that are being tested, again, with the same information that we needed before. We do need all these 2 blood samples. We will detect the variance down to the 0.1%, and we have high value content, in which we can see KRAS, NRAS, BRAF mutations that you all know, as I mentioned in the FFPE tissue assay, that are of great relevance when testing for this kind of cancer. In the case of breast cancer, we do also have a huge and good panel, that is the breast cancer panel for liquid biopsy, in which we include 12 different genes, once again of great relevance in this kind of cancer. We have the PIK3CA gene, the AKT1, ESR1, etc. As you know, this will be a great way to know if your patient has developed cancer even before images are able to tell it. And last but definitely not least, when it comes to liquid biopsy, it's the one that we all know, and it's a great example of why liquid biopsies are so important, and that is lung cancer. When it comes to lung cancer, sometimes it is of great importance how difficult it is to obtain a sample from that patient due to the risk that you put your patient in. So in this case, we are going to be testing for your patient in a minimal invasive way that might help them with the treatment, and that's how we see different variations in EGFR, KRAS, BRAF, etc. Genes that you know are of great relevance when it comes to knowing how your patient is evolving in the case that you're treating them, and how it is better to treat them in the first place. And that's everything we have on our somatic department, but we do have some other options that I wanted to talk to you about, and that's the hereditary cancer syndromes. As you know, between 10 and 15% of diagnosed cancers are caused by inherited oncogenic mutations, and these mutations, even though they are not as frequent, we know that are of great relevance to be able to detect and control these patients in case they will have this higher risk of developing cancer in their lifetime. So we do offer two different tests. There are the Clarity Test Plus and the Clarity Test, in which we do include 2000 genes or 39 genes. And it's another way of getting to know how your patient will develop, to know if you will need to treat them in the future, and just to be very focused on that patient and know what to do with them. So that will be all that I have to say to you today. Thank you once again for all the attention that you just gave me. And if you have any questions, I will be glad to answer them. If any questions were left without answer, Luis will be glad to answer to you. And if he didn't have the answer, he could tell me anything. I will also be grateful to talk to you again. So if there are any questions, now's the moment. Thank you.
So thank you very much, Vanessa, for the nice presentation. We do have some questions that I've been gathering from the chat. We will address the questions, but you are free to still write down more questions if needed. I tried to organize them from more general to more specific topics. So let's start with this one. At what cancer stages are these tests typically requested?
Okay, so that depends on the clinician and on the test that you are talking about, but it's principally to know what to treat your patient with. So that would be a great stage to test them. But if you want to know, for example, with liquid biopsies, as you know, are many times used to assess progression with different treatments. So it depends on the kind of test that you want and the information that you want to gather from it.
Mm-hmm. Okay. Then we have a long one. I try to read it. For the different pan-cancer tests, will the analysis and report be adapted to the tumor location, organ, or will it be exactly the same report regardless of the tumor origin?
No, of course, we do have a personalized test for a personalized test, not but a personalized report, and we do focus on the different mutations that are the most relevant according to the tumoral origin, always taking into account if it is metastatic or not, and if we have to take into account the origin of it and to know the best options for the patient. But we always have the possibility of you talking to us and just trying to get around the possibility of assessing for you most want of it.
Mm-hmm. Okay. So you please know we have this flexibility in the reports.
Yes.
Then we have a more specific question on HRD. So if I have a patient with primary ovarian cancer and my main objective is to determine if PARP inhibitors will work, is the HRD test sufficient, or should I order the pan-cancer comprehensive assay plus with the HRD?
Okay, so if the only information that you want is to assess the PARP inhibitors way of action, if they will work on your patient, it would be fine ordering just the HRD test. But in the case that your patient does not respond to HRD and you want more information, you will have the pan-cancer comprehensive assay plus HRD. I think that would be a great option too. So it just depends on the objective of your study.
Mm-hmm. All right. Then some practical questions. How should we prepare the samples if we can't send the whole block, but some slices?
Okay, so the most important thing is that we want the slices to be sent on any tube, like an Eppendorf tube, for example, but we do not want them fixed on a slide because it does make more difficult the extraction, and we can lose some of the material along the way, so we do not want that. So just by sending some slices on an Eppendorf tube, we will be able to extract the DNA and the RNA and just help you with that. But if you have any other questions, for your specific case, we will be glad to answer it.
Okay, here maybe we should remind the minimum number of slices we usually need?
Yes, we usually need 2 to 3, but it all depends on how much tissue we have available on that tumor.
Sure. Okay, and then I'm checking that we have another one here regarding the turnaround time for the generation of the reports, in particular for the pan-cancer comprehensive assay plus with the HRD.
Okay, so the comprehensive plus the HRD is one of our biggest reports. So we do take around 3 weeks, 21 days, 21 natural days from the moment that the sample gets into our installations, but we always try to adapt to and make them faster just so you can treat your patients as soon as possible.
Very good. And I'm checking that we don't have further questions actually. So I think if that's no more questions from the audience. So I think we can wrap up. Thank you very much, Vanessa, again for your effort and your presentation. And thank you very much to the audience for the time with us today. Like Vanessa said, we are happy to answer further questions via email or phone. Of course, I will personally rely on Vanessa as our head of oncology, our expert here. And don't hesitate to present your clinical cases and we will try to support as much as possible.